welcome back. So we're going to finish up chapter 20, which is all about antimicrobial drugs. I apologize for the interruption and cutting abruptly the last video, but I'm sure you could hear I had a kitty that needed me. Okay, so now when we talk about inhibitors of cell wall synthesis, these are all drugs that are going to lead to cell death, so we call them bactericidal. We're targeting different targets of the cell wall synthesis, but what you'll notice is that both the beta-lactamarine antibiotics and the polypeptide antibiotics are they're interfering with the ability to make those cross links, those peptides that hold together the chains of carbohydrate, the NAG and NAM subunits. So by interfering with those peptide cross links, there's nothing to hold the layers of the carbohydrates together in place, and then it becomes weak, and it gives out under osmotic pressure, and the insides of the cell contents will leak out. And the cell could completely lyse open, burst open, which I have an image of that right here. So you can see this hole has been formed because the weak cell wall. And here in this cell next to it, you can see it's the cell has flattened. It's getting multiple holes, and the internal contents have leaked out. So the bacteria cell has lysed open due to penicillin antibiotic that weakened the cell wall. So now we want to see, well, what is that mechanism? So penicillin and some of the other antibiotics that have a particular structure called the Beta-Lactamarine drugs. If you look here, this pink structure is the Beta-Lactamarine. The Beta-Lactamarine binds to the active site of the bacterial enzyme shown in green. The bacterial enzyme is the peptidase that normally would link these peptide chains together, which then holds the NAG-NAM chains together, right? So normal bacterial enzyme makes that final bond between the peptides, so that peptide bond, and that is what gives the cell wall its ability to be strong and hold together. The beta-lactamarine drug will bind to the active site of the enzyme, and then it's not able to make those peptide bonds. So these are all separate and weak. Okay, so we have natural penicillins that have the beta-lactamarine. We have penicillins that we've modified, so they're semi-synthetic. They have a natural portion and a synthetic portion that have been linked together. And this can be, semi-synthetic can be to make it more broad spectrum, right? Extended spectrum, which is what we see with the amoxicillin and ampicillin. But we also have some semi-synthetics, which I'll show you here, that remain narrow spectrum, but they become resistant, and we say it's resistant to penicillinase. Another word for penicillinase is beta-lactamase. So beta-lactam is the ring, and ACE would be an enzyme that destroys the ring. So what's happened is once we introduced these beta-lactamarines to bacteria to fight bacterial infections, some bacteria were able to make an enzyme to destroy it, and there began antibacterial resistance. Okay, so then we in turn tried to make a different version of the drug that the bacterial enzyme would not be able to destroy. And so you can see that all of the different versions of the beta-lactam drugs all have this what we call common nucleus, where you have this beta-lactam ring here, and then you have another ring right next to it. And so this whole region here is consistent through the different drugs. And then on cephalosporin, we see it's even more modified, where you pretty much only have the left half of the common nucleus. The ring to the right has been expanded, it's larger, and there are more side groups on it. So that part is very different than what we're seeing up here for the semi-synthetic penicillins. But cephalosporin is a beta-lactam. So it's not so cephalosporin is not a penicillin, but it's in the same family because it still has the beta-lactam ring, which ultimately will have the same reaction of what we see here, right? A beta-lactam ring binding to the peptidase enzyme responsible for cross-linking the peptides in the cell wall. Okay, so you need to know beta-lactam ring antibiotics include natural penicillin, semi-synthetic and extended spectrum penicillins such as amoxicillin and ampicillin. We have another group that I haven't talked about yet, the carbapenems. These are broad spectrum, and they're actually newer, and they're pretty effective. And I think they are being reserved so that we don't get resistance against them, and we'll have something available when needed. And cephalosporin, which I mentioned in the last video, there are different generations, and depending on which generation, that will determine exactly which group of bacteria it kills. So it is an extended spectrum, but it's still a relatively narrow spectrum because you will either target gram-positive or gram-negative or Mycobacterium, but none of the generations are going to get all three of those groups, okay? Different generations of cephalosporins have different range of activity or spectrum. Some are better for gram-negative, but then they lose their gram-positive effectiveness. And so these are somewhat narrow spectrum that shifts either to the right or to the left, depending on the generation of the drug. Okay, now going back to the semi-synthetic and extended spectrum penicillin, semi-synthetic and synthetic penicillin will either increase the killing range, right, extended spectrum, or become resistant to the bacterial enzymatic destruction. So again, remember that the semi-synthetics that I'm not asking you to memorize, but we do see here on this slide here, they have been modified so that they don't get destroyed by the bacterial enzyme, which sometimes that bacterial enzyme is called penicillinase, and other times it's called beta-lactamase, and that's the same enzyme. It's an enzyme that destroys the beta-lactam ring and makes the drug not work anymore. We in turn modified the drug to be resistant against that. However, we are not able to do both. So if we want to extend the spectrum and have it work against both gram-positive and gram-negative, we can modify it to target both positive and negative gram bacteria, but then it will still be susceptible to destruction if the bacteria have penicillinase or beta-lactamase enzyme. It can still destroy cephalosporin. Okay, so antibacterial resistance is due to the enzyme penicillinase, which is also called beta-lactamase. Okay, and it's very common. Most of the bacteria these days will have penicillinase or beta-lactamase, okay, which again cleaves that beta-lactamarine, and then this no longer is able to do its job. Okay, this slide here, and I do apologize for flipping back and forth on these slides. This slide here, I do not need you to memorize. I simply wanted you to see that there are different types of penicillin. They all have the common nucleus, but they have slight differences on the side, and this will depend on if it's an injection or if it's a pill that you take orally. And that also changes the concentration in the body and the duration in the body. So pills will give you a high concentration that wears off very, very quickly, right, within about 6 hours typically. And that's why antibiotics are usually taken 3 times a day. Some of them are taken 4 times a day. And you usually take them for 7 to 10 days to ensure that you kill all of the bacteria and don't end up with resistant bacteria. It's still possible. So overall, even if you follow the instructions and take all of your antibiotics, you still don't want to take antibiotics unless you absolutely have to. Okay. But again, don't worry about memorizing these specific types of penicillin. I'm not going to ask you which one's injectable, which one's oral, nothing like that. Okay, there's also a chart graph in your book about the amount of times and concentration of the different types of penicillins, and I'm also not going to ask you about that. There's so many drugs and modes of action to memorize in this chapter, so I'm just trying to stick to the bare minimum of what you need to know. Okay, so now if you go back to this, there's a really great video. If you only watch one video in this chapter, make it this one. This starts off explaining the cell wall synthesis, the inhibition of cell wall synthesis by beta-lactamarine drugs, and then it also will then go over the different mechanisms of antibiotic resistance, which you'll need to know. So it's a great video, and it's not very long. It's pretty straightforward and to the point. Okay, so remember, cell wall, if you inhibit cell wall synthesis, it's going to become weak and the cell will lyse open and die. Polypeptide antibiotics are doing the same thing but with a slightly different target, and this is vancomycin and bactricin. So let's go ahead and take a look at that. So vancomycin is actually binding to the polypeptide and blocking the enzyme from being able to cross-link. So instead of binding to the enzyme, you're binding to the substrate. Okay. Otherwise, so a very slightly different mode of action, right? Different drug target, but you're still blocking the same overall activity, right? Of these you're preventing the cross-link of the polypeptides. This is the drug, quite huge, okay? Vancomycin, a huge molecule, okay, that will bind to specifically the D-alanine amino acids in the polypeptide chains, and then that will prevent the bacterial enzyme, the transpeptidase enzyme. So same enzyme that we saw with the penicillins, but instead of binding to the transpeptidase directly, you're just being a competitive, non-competitive inhibitor, where you're binding to the substrate instead of the enzyme. Okay, so these are called glycopeptides. They're bulky, and because of this, they're not able to pass through those poron channels on gram-negative bacteria. So these are only effective against gram-positive bacteria. Okay. Also, because they're bulky, we do not get very good intestinal absorption. And so there's two caveats with that. If someone has an infection of their intestines, such as C. diff, then this would be a really good drug to give them orally because it will stay in the intestines and take care of the infection, right? But if someone has an infection elsewhere, you would need to give this by IV, intravenously, because it will not be absorbed through the gut, right? It'll get trapped in the intestines. So you would only take it orally if you're trying to treat an intestinal infection. Any other type of infection, you would need to give an IV administration of the drug. Okay. I attached two very short videos that show the mechanism of vancomycin. And this is actually an image from one of those videos that shows you that this drug will bind here, and then the transpeptidides won't be able to come in and make that bond. And so then these two chains of NAG and NAM carbohydrates will not be held together. So the same end result as penicillin. All right. The IV form of this drug can cause some side effects that have to be monitored closely. It can be cause some renal toxicity because the kidneys are filtering it out of the blood. And this is referred to as red man syndrome. Okay, it's not necessarily fatal, but it is a complication that needs to be monitored closely and dealt with to make sure that you don't end up damaging the kidneys. Okay, this drug is mainly used to treat MRSA, methicillin resistant Staphylococcus aureus, and as well as some other infections like listeria. It's good at treating streptococcus, enterococcus, and pneumococcus. So this vancomycin is reserved for these pretty serious infections. But as you've seen already, I've talked about that there are strains of Staphylococcus aureus that are resistant to both methicillin and vancomycin. We call those multi-drug resistant. And there's others that are resistant to vancomycin but may still be susceptible to methicillin. Okay. So again, always need those cultures so we can find out what the infection will actually be sensitive to and prescribe the right antibiotic. So it is pretty common these days to be started on an antibiotic and have to switch to a different antibiotic once the results of the culture come through. Okay. Now we have another class of antibiotics called inhibitors of protein synthesis. We have the aminoglycosides, which they are broad spectrum, but they have some really nasty side effects, so they're really not used often. They are really reserved as a last resort when there's nothing else you can use, such as streptomycin for tuberculosis or gentamicin for Pseudomonas aeruginosa. And remember, Pseudomonas aeruginosa is particularly challenging because it has that rapid efflux pump. So there are not very many drugs that work against Pseudomonas aeruginosa. Tetracycline is a pretty popular drug. Tetracycline does cause weight gain, and so for this reason, it's been abused by the food industry for getting animals like chickens and cows, livestock, right? Getting them to bulk up faster so they can sell them faster and make more money. This has led to a widespread resistance of tetracycline antibiotics. So we really need to change the food industry And other industries that use antibiotics in their materials, you see this a lot in pet products, baby products. There's these like microban products and other similar products that will say antimicrobial this or that. And we really need to stop those. All of these different uses are bad uses of antibiotics that lead to faster and more widespread resistance. Same thing with antibacterial hand soaps and things, right? The hand sanitizers are alcohol based, they're not drugs. We think that it's okay. I still advise against it. Soap and water is all you need to effectively wash your hands, and then dry them on a clean towel. So it could be a disposable paper towel, or it could be a thinner towel that you know is going to dry very well and quickly, or you are changing your hand towels often, and that's it. So you don't need to be using harsh chemicals or valuable drugs. Okay, so a little bit more about tetracycline. It has a very high affinity for bacterial ribosomes, which is how it blocks protein synthesis. It's also really good at tissue penetration, so it makes it a very good drug for difficult infections like UTIs and respiratory infections. It's also good for treating intracellular infections such as chlamydia and the rickettsia bacteria. So this drug will interfere with the tRNA being able to bring in amino acids to the ribosome. So it's blocking protein synthesis. And but so it sounds like this wonder drug, right? It's broad spectrum, it interferes with protein synthesis, it's really good at penetrating body tissues, at getting these difficult infections. But because it's broad spectrum, and this is true of any broad spectrum drug, it is much more likely to kill off your part of your flora, your normal flora, and make you susceptible to opportunistic infections, so the overgrowth of the flora that are not killed. So it's very common to get what we call a super infection with yeast or C. difficile, right? So it could be different locations of the body. It's not always intestinal, but of course our intestines are going to typically be the most susceptible when you're taking oral antibiotics. So if, for instance, if a female needs to go on tetracycline, she's usually also prescribed something for a vaginal yeast infection to prevent it from occurring. They may encourage supplements like probiotics to help with both the intestines and to decrease the likelihood of vaginal infection. So men, of course, can still have serious intestinal infections if they're on tetracycline. So it's also not something that should just be prescribed lightly. But if you do have a UTI, especially if it has gone to the kidney, this would be a good drug for that. Okay, now another group of protein synthesis inhibitors are called the macrolides. These are really cool. I showed you the what the drug looks like because it's a star-shaped ring, which is just really pretty and fun, right? So the most common drugs in this group are erythromycin and azithromycin. And these are narrow spectrum for gram-positive bacteria. They also interfere with tRNA and block protein synthesis. And the azithromycin has been made to be a little bit broader spectrum. It also has a little bit better tissue penetration. So just like we saw with the tetracycline, the azithromycin is going to be given for those more difficult infections like UTIs and respiratory infections. There's a little video here to show you the mode of action and talk a little bit about the macrolides. Okay, it's a very good alternative to penicillin. Right, penicillins also target gram-positive bacteria. Okay, so this is a review of those 3 drugs. Streptomycin, which is, I know I didn't write that, but I did. I wrote it here. Streptomycin is one of the aminoglycosides. So the aminoglycosides, such as streptomycin, are going to target the 30S portion of the ribosome and block the mRNA and tRNA interaction. Okay, so it actually causes the mRNA to kind of be warped, kind of change its shape a little bit. It's going to be read incorrectly, and then you're not going to get the correct amino acids coming in, and you'll have a non-functional protein. Okay. On the other hand, the tetracyclines are going to interfere with attachment of the tRNA into the ribosome, and so you're not going to get any synthesis at all. Okay. And then the macrolides up here in the top left corner, we see there's this tunnel where the growing polypeptide comes out of the ribosome, and it's going to block this tunnel. So it's going to lock up the whole process, and it's going to stop the process through this tunnel. So the drug binds in here. Okay. So those are the 3 modes, mechanisms for the inhibition of protein synthesis. Just make sure here under streptomycin that you remember that that's under the branch of aminoglycosides. Okay. I couldn't fit it. It was too big of a word to fit here. Okay. Rifamycin is a nucleic acid synthesis inhibitor. It inhibits mRNA synthesis, and remember, mRNA is needed so that you can then have translation occur, and so just a little bit sooner than what we just looked at, right? It also has really good tissue penetration, and we can even see that it can treat infections of the cerebral spinal fluid, so central nervous system, and even abscesses, which are some of the most difficult to treat. So really good for these difficult infections. And we primarily use this to treat tuberculosis. So this is one of the first drugs that we would try to treat tuberculosis with. Okay. I do not have an image here for you for rifamycin. You just have to remember mRNA synthesis inhibitor. For the fluoroquinolones, I do have an image. It's right here. And so they're shown as being these little triangles, and they're binding to the DNA gyrase topoisomerase complex. And so it's labeled here as topoisomerase in the image. And then in your textbook, it calls it DNA gyrase. So it's actually it's a complex. So it's both. It's topoisomerase, DNA gyrase. Okay. And these can be quite nasty drugs, really bad side effects. So they really should only be used when there's no other option. Okay. They can be very, very effective at treating UTIs and pneumonia. So when these infections are not getting treated, like you've tried another drug and it didn't take care of it, or someone has a very serious life-threatening infection and you don't have time to try alternative drugs, that's when you bring out things like the fluoroquinolones. They can have side effects such as bursting, rupturing tendons. Tendons, as you know, hold your bone and your muscle to your bone, rather. And so that's a terrible side effect. There's also other side effects that are kind of debated with neurological side effects, and so it can be pretty bad. There's a short video here for you to watch if you want to. So that concludes, it's just these 2 examples I'm giving you for nucleic acid synthesis, right? So the fluoroquinolones are going to block the DNA from being unwound, so you're not able to replicate it, because now there's the two strands are going to be stuck together, so your replication is going to stop. So it blocks DNA replication, whereas rifamycin is blocking mRNA synthesis. Okay, so two different modes of action. Okay, so injury to plasma membrane, we have the lipopeptides such as daptomycin. This is only for gram-positive bacteria, and it structurally changes the membrane. And then, so step one, daptomycin binds and inserts into the cell membrane. Okay, so we kind of see that it binds and then it inserts into the membrane. Then it aggregates the cell membrane, so it aggregates into these small clusters of the drug, and then it will lead to an alteration of the shape of the cell membrane and eventually lead to a hole. So here you see that it's opened up space between the lipids. So now there's a hole, and you can see this exchange where you normally keep a very high concentration of potassium inside the cell, but it allows it to leak out. So when you let these ions flow, you no longer maintain the correct membrane potential, and you lose your ability to for the cell to perform its normal activity. And this is primarily used to treat MRSA, methicillin-resistant Staphylococcus aureus. Okay. Then we have competitive inhibitors of essential metabolites. And remember that bacteria will make their own folic acid. We don't. We have to consume folic acid. We do not synthesize our own folic acid. So that's why this drug is able to target bacteria and not us. So there's this precursor, PABA, and I think I've shown this to you before. But PABA is a precursor to folic acid. Folic acid is a coenzyme needed for processes like DNA synthesis, mRNA synthesis, protein synthesis. So all of these anabolic processes of building things that the cell needs, you're going to need the folic acid coenzyme for the different enzymes in these processes, in these metabolic pathways to be able to work. So you block the metabolic pathways, and then you will inhibit the bacteria. They will eventually die, but what it turns out is that you slow them down enough for the immune system to take care of. If the immune system was really slow, then this would eventually kill the cell, but we don't consider it bactericidal. We consider it an inhibitor. Okay, so sulfa drugs inhibit folic acid synthesis. And then by inhibiting folic acid synthesis, you're inhibiting the metabolic pathways, right? This is considered broad spectrum, and it's really good for UTIs and for burn patients, to prevent infections in burn patients. Yeah, that's all I wanted to say about that. All right, so now we'll talk about drugs used for different types of infections. So everything we've talked about up to this point was for specifically talking about bacteria. Now we're going to talk about everything else. Okay, so fungal infections, remember when you get to the eukaryotes, they're much more similar to our cells. So we have very few targets, unique drug targets. One unique target would be the fungal cell membrane sterol. So we have cholesterol. Mammals, including humans, have cholesterol in the cell membrane. Fungal cells have ergosterol, and ergo is you'll see sometimes as a root word for fungal things that are related to fungi. Okay, and so the membrane becomes excessively permeable and the cell dies. Okay, so the mode of action for all of the antifungal drugs we're going to look at today will be the same, targeting the same sterile fungal cell sterol. So entering the plasma membrane and causing it to leak its insides, right? Become leaky cells, and that will cause them to die. Okay, so we have polyenes, azoles, and alliamine. Okay, so polyenes, our most popular drug is Amphotericin B. This is typically given through an IV. This is going to be patients that are hospitalized. They have a systemic infection. Most of these infections begin as a respiratory infection. And in normally healthy individuals, their immune system would normally handle that. But because the population of immunodeficient individuals is increasing, so we have more people with diabetes type 2, more people with AIDS, and other reasons for being immunodeficient, they're not able to get those fungal infections under control, and they become systemic. This third one should say blasto. I think my word processor kept changing this, but it should say blastomyces, BLASTOM. And we'll see this when we do our section on fungal infections. We'll talk about each of these and a few more examples. So don't worry about knowing these diseases, but just start to learn their names, right? So Amphotericin B is used for systemic infections with the fungi called histoplasma, the fungi called coccidioides, and the fungi called blastomyces. Okay, and this drug can be hard on the kidneys, but that's a risk we're willing to take when someone has a systemic life-threatening infection, right? Okay, so the next category, the azoles. Some of these are over-the-counter, non-prescription, and but we do have some that are still prescription. And they're usually for topical use for cutaneous fungal infections, mycoses. Mycoses means fungal infection. So things like athlete's foot, jock itch, or a vaginal yeast infection. You can go to your local drugstore, or even I think probably most regular grocery stores probably carry this too. And you can get that topical cream and treat it yourself. And I think it usually takes a long time, like 14 days, I want to say. All right, so then we have the Anti-Protozoan Drugs. And these protozoa can be even more difficult to treat, but they're not as common infections for people. So you'll start seeing in your book, it's going to start talking about drugs that are mostly used for dogs or horses, right? Different animals, because it's more common for the animals to have serious infections with these than it is for people. But because it is common among animals, you could contract it from contaminated water. That originally it's contaminated from the animal that's infected. Okay, so mosquito-borne infection with Plasmodium causes malaria. So this would be from mosquito bites, and you are probably familiar with malaria at least a little bit because it is such a huge problem that we've been trying to get under control for a long time. And there's a drug, Chloroquine, that inhibits DNA synthesis. However, this was used so much so that most areas where malaria is endemic now have resistance to the drug, and so it's no good anymore, which is tragic because it's easy to make, it's cheap, it's affordable, we're able to distribute it to all these communities, but it's really not helpful anymore. In fact, the main way that we're trying to fight malaria is by teaching people how to avoid having mosquitoes, such as getting rid of any water that's just sitting. So don't have any sitting water around your residence. Avoid going out at dusk and dawn when mosquitoes are most active, and sleeping with bed nets to prevent mosquitoes from being able to get to you while you sleep. Okay, so that's the main way that we're fighting against malaria these days. We do have a few other drugs, but they're expensive, and so it's not really helpful for most people. We have another, so Plasmodium, I just want to clarify. Plasmodium is the protozoan parasite. It's transmitted by mosquitoes, and the disease that it causes is called malaria. Okay. Metronidazole is another drug that can target quite a few protozoan infections, such as trichomonas. Commonly causes vaginal infection. It can be sexually transmitted, but it is not strictly a sexually transmitted disease. Giardia lamblia is a common serious intestinal infection from drinking water, such as like you go camping and you drink fresh water from a stream, and you get this infection. So it used to be called hitchhiker's diarrhea, because hitchhikers would just drink from whatever fresh water source they could find. It was also called camper's diarrhea because people going camping will drink the water nearby. So this is why when you go camping, you have to have some type of water filtration, water treatment, or you have to bring a lot of water with you, or you need to boil the water, right? So that you don't get this. Amoebic dysentery. Amoebas are also protozoans, and there are specific types that can cause dysentery. Remember, dysentery is bloody diarrhea. Okay, so there you go. Antiviral drugs. So we're going to talk about a few different categories of antiviral drugs. But it's important that you know that antivirals in general don't work very well. So if someone has a serious viral infection, such as HIV, they're going to be given what we call a cocktail of antivirals that have different mechanisms of action, and that are also known to not interfere with one another. And this is how we've been able to prevent the development of AIDS for a lot of HIV patients. It's by taking at least 3 different antiviral drugs. So antiviral drugs that specifically target HIV are called anti-retrovirals. Because remember, HIV is a retrovirus that goes backwards. So HIV is the only human retrovirus, and it has 2 unique enzymes. Integrase, which allows it to insert the viral DNA into the host DNA. And then it also has reverse transcriptase to make that DNA. So HIV actually, when you have the actual virus that infects the cell, is already RNA, but it uses the reverse transcriptase enzyme to make DNA, and then it uses the integrase enzyme to insert that DNA into the host. It's kind of bizarre. It's actually very bizarre, because the only virus that has, in fact, it's the only thing that has reverse transcriptase. None of the bacteria, none of the protozoa, no plants, nothing that we know of has reverse transcriptase other than the retroviruses. Okay. So anyways, we have all sorts of different antivirals to target HIV, different parts of the viral life cycle, such as binding and uncoating, and the unique enzymes integrase and reverse transcriptase. So that's my talk on HIV for now. Other antiviral drugs we'll look at, okay, try to keep separate from the anti-retrovirals. Okay, so again, antiviral drugs are very difficult to make, and for this time, the reason is because the virus is hijacking our cell. It's using all the parts of our cell to replicate itself. So it is using the host cell machinery to replicate its nucleic acid to produce and assemble the viral proteins. Okay, and then it will even take a lipid coat from the cell's own lipids. Okay, so if it's a membrane virus, a virus is a membrane. Not all viruses have membranes. Okay, so we have interferons, which is a drug that we actually make ourselves when we have a viral infection, and it's considered a cytokine because it's a cell messenger. It's a little chemical. It's released from the cell that tells the cells around it that there is a virus, and to shut down the processes that would allow that virus to infect it. So basically, it's too late for me, but save yourself signal. Okay, and we call that alpha interferon. It's really, we've learned how to make interferon as a drug that the person can take, and this has been very helpful in fighting viral hepatitis. So hepatitis B and hepatitis C in particular, right? We have a vaccine for Hep A and Hep B. We do not have a vaccine for hepatitis C. Okay. Furthermore, not everyone gets vaccinated, or they don't get all of the booster vaccinations, so they can still get hepatitis, right? If they don't complete their full series of vaccinations. Okay, so that's one way to fight viral infection is with interferons. Then we have antiviral drugs that target the assembly or the exiting of the virus life cycle, and we call these protease inhibitors. So you're inhibiting the enzyme that's putting together proteins. So protease is an enzyme that cleaves proteins, and so a lot of times what happens is that these viruses make like one big thing that they cleave into small pieces and assemble. And so if you remove its ability to cut them into small pieces, then they cannot assemble. Sometimes that's also how they exit. They have one receptor that they're bound to the cell, and then they need to cleave it. And if they can't cleave it, then they're stuck to the cell and they can't go spread to other cells. Okay, so influenza is commonly antivirals that treat influenza are usually protease inhibitors. Again, they're not super good, so a lot of times when you have influenza, you're just told to get lots of rest and drink lots of fluids, right? And maybe take some Tylenol, okay, or treat your symptoms. But if you have a very severe case of influenza, then you might be prescribed an antiviral to help your immune system, but it's not a miracle drug, okay? Nucleic acid synthesis inhibitors are a little bit more popular because they can be used for herpes viruses. This is predominantly genital herpes, also hepatitis B and C, and eye infections from cytomegalovirus. Cytomegalovirus we'll talk about when we get to our virus chapter, but this is a virus that causes lots of cells that are next to each other to basically become one super large cell, and so it's mega, right? Mega cell. So cyto for cell, mega, right? It's going to make a huge mega cell. So that's its unique name. Okay, So acyclovir is one of the most popular antiviral drugs used, and there's a lot of derivatives of it. And I'm not going to worry about all those names, just know acyclovir. Okay, and it has a lot of derivatives, and they pretty much all function in the same way. They're a nucleoside analog. So what happens is the virus has a kinase that it uses to phosphorylate and then insert into the nucleic acid that it's making to copy its own genome. And so this will have no effect on cells that are healthy with no virus infection, because they won't have the viral kinase, right? Only the cells that have the infection will have this analog get inserted instead. Okay, so acyclovir and its derivatives are nucleoside analogs, which will get phosphorylated by the viral kinase, and that's called a thymidine kinase, and then it gets incorporated into the DNA or RNA that's being synthesized. Again, that kinase is a viral kinase, so only under viral infection will the drug have any action, okay? And there will be zero effect on healthy cells, okay? All right. And so this shows you how it's very similar. It has the same kind of core structure as the deoxyguanosine, which is the guanine nucleoside. And so it can get utilized instead. But this it makes it'll actually stop the synthesis because it won't allow you to go to add on the next nucleoside. So it halts. So we have an image of that here. So normal thymidine kinase in a healthy cell is going to take phosphate and the normal nucleoside and put them together, and then the DNA polymerase is going to take that and incorporate it into the growing DNA. But when it has the viral infection and you have the drug acyclovir, the phosphate and the acyclovir get put together by the viral kinase, and then it gets taken up by the DNA polymerase, and then it blocks it. It can't move past that step because it's not actually the guanine nucleotide that it thought it was. And so then DNA polymerase gets blocked, and then the whole synthesis gets stopped. Okay? All right. So now we can talk about resistance to antimicrobial drugs. And we already started talking about resistance with the enzymatic destruction, right? So so how does this originate? Okay. Some bacteria in a population will just naturally have resistance. You have to remember that a population of bacteria, there's millions and millions and millions of bacteria. And over time, as cells replicate, these bacterial cells replicate their DNA to divide, they'll make mistakes. And sometimes those mistakes will lead to new protein products. Most of the time, it doesn't lead to anything. Most mutations are non-functional and even harmful, but occasionally they are beneficial. And so you'll get this natural resistance. So you take an antibiotic or you use it in some way and you kill the majority of the population that is susceptible. But the few bacteria that have this natural resistance are going to survive, and they're going to continue to grow and multiply, and eventually you'll have a whole population that are resistant. So this is the first step in antibiotic resistance. Once you have a population of resistant bacteria, they can share this resistance gene with other bacteria that don't even have to be the same species. And they can do this very rapidly through conjugation. We've talked a little bit about bacterial conjugation way back in chapter 4, and we'll talk about it again next week in more detail. But bacteria that have that sex pilus, if they have the genes to undergo conjugation, they can copy their plasmid that contains the bacterial resistance genes and just transfer it right over to the bacteria that it decided to grab onto. And now that bacteria has that plasmid, which now it's going to make this product and it's going to become resistant to that antibiotic. Sometimes plasmids will have resistance to multiple different drugs. And so this is becoming more and more scary as there are more and more infections each year that we cannot treat. We already have hundreds of thousands of people dying from antibiotic resistant infections. Okay, so this conjugation, bacterial conjugation, is a type of horizontal gene transfer that leads to the rapid spread of antibiotic resistance. Whereas when you're talking about parent cell dividing, that's a vertical transfer. Okay, so resistance always starts out from just a natural resistance, but then it can then be spread through conjugation. Okay, so what are the modes of antibiotic resistance? Or what other factors contribute? First, I'm getting ahead of myself. So we're looking at ways that we as people are contributing to this issue. So antibiotic resistance, as I said, is natural, but it is accelerated the more we expose populations of bacteria to antibiotics. And so the misuse of these drugs is going to select for the resistant mutants, right, the bacteria that naturally have resistance, and it's going to accelerate the spread of the antibiotic resistance. Okay, so the common term is antibiotic resistance, but really it should be antimicrobial drug resistance, because we know that most of the drugs are not true antibiotics, but this term is misused in medicine and popular, reference. So a lot of times we just go, we're defeated and we go with what's the popular terminology instead of the correct. All right, so misuse of the drug selects for the natural mutants. They grow and replace the population that you killed off, and now you have populations of resistant bacteria, just like we talked about in the last slide. So what is misuse that leads to this selection? It can be using outdated or weakened antibiotics. So this, you have to remember, we're talking global, not just within the United States. And there are a lot of countries that don't have any regulation on antibiotics. Anybody can buy them, anybody can sell them, and you don't know what you're getting. And you don't necessarily know which antibiotic you need because you're not a doctor and you don't have any lab work to confirm what you need. So the misuse of antibiotics is huge outside of the United States. It's overwhelmingly huge. But even within the United States, there are doctors who they will give in. If the patient wants an antibiotic, they'll give it to them even if it's not a bacterial infection. Okay, so antibiotics given for the common cold or other inappropriate conditions is unfortunately pretty common. And then patients that need it, they're given the correct antibiotic, they still might not take it correctly. And so things like not completing it is a huge problem. People giving their family and friends their leftover antibiotics when they're sick is a huge problem. You should never do that. How do you know which type you need? And if someone already took some of them, it's not going to be enough for your problem. Just nothing about that makes sense, okay? You should always go to a physician, see if you actually have a bacterial infection, know what type of infection you have, what type of antibiotic you need, based on your weight, and other factors, they'll give you the correct dosage and period of treatment. Okay. All right. So we see the initiation of antibiotic therapy. You start to rapidly decrease the amount of bacteria in the patient. At about 4 days, you will get the least amount of bacteria in the system. Now, this red line is showing antibiotic resistance of bacterial population. At this point, you're killing off all the susceptible bacteria, or most of the susceptible bacteria, and now you've selected for the resistant bacteria. So the resistant bacteria are going to start to take over the population, and they'll continue to grow and grow and grow because they're resistant, right? They're not affected by the antibiotic. So now you have a large amount of resistant bacteria. Okay. So the development of antibiotic resistance mutant during an antibiotic therapy. So this is why I said even if you take the correct drug for the correct amount of time, you can still be selecting for antibiotic resistant bacteria. Now, it's possible that these resistant bacteria are not the same bacteria that are causing your actual illness. So you may feel completely better and not have any consequence of this population. This could just be one of your normal flora that now are resistant. But if at some point in the future that group maybe is an opportunistic pathogen and you end up with an infection down the road, then it's going to be very difficult to treat because it's resistant. Okay. All right, strategies or mechanisms of antimicrobial resistance. So bacteria or other pathogens might block the entry of the drug. So the example of this would be the Pseudomonas aeruginosa, I have a category for rapid efflux pumps separately. So blocking the entry of the antimicrobial drug could be something like the receptor that's used to get inside of the cell gets changed, or some other strategy to block the entry of the drug. Okay. Enzymatic destruction of the drug we already saw with penicillinase, also called beta-lactamase, right? And other bacteria can make other enzymes that target other drugs in the same fashion, right? You destroy the drug. The rapid efflux pump of Pseudomonas aeruginosa quickly removes the drug before it has time to do what it's supposed to do, right? And then there's alteration of the target site. So maybe you don't have an enzyme to destroy the drug, but you change where the drug is supposed to bind and have an action. And so the drug no longer has a target and it's useless. Okay. Again, that video way back in the beginning on the beta-lactam discussion, use that video. It will review all of these antimicrobial resistance mechanisms, as well as your textbook also elaborates. Okay, so those are the 4 mechanisms of antimicrobial resistance that I want you to memorize. All right, now sometimes we can combine drugs and get a better outcome, and other times if you combine drugs it has a worse outcome. So synergism is the term for 2 drugs that you can take and have a greater effect than either one alone. So you might say, why not just have a bigger concentration of drug A? Well, maybe having a larger concentration of drug A starts to produce negative side effects. But if you have a small amount of drug A and a small amount of drug B, you get the same really good treatment without the side effects, right? So that's one possibility. So synergism just simply means that the 2 drugs work better together than either one is capable of alone. Antagonism is when you have 2 drugs that interfere with each other, and so they become less effective than if you were to take just one of them. And so here's an example of synergism where you can see a drug, amoxicillin, and you can see this other drug here, and they each have their zone of inhibition, right, individually. It's a little hard to see. It's not really the best image, but you can see that they kind of have this zone of inhibition, each of them individually, but where the diffusion of the drugs combines, you get an even bigger inhibition. And so I'm sure you could probably find better examples of synergism on a plate than what's being shown here, but that's what I had to work with for this time. Okay, so 2 drugs work better together than either one does by itself. Future of drugs, I actually took out, but I forgot to take out this slide. So go ahead and cross this out. There is a cool section that I could talk about for possible future treatments, which is pretty exciting and interesting. But like I said, I know you already have a lot to memorize for this chapter and the previous chapter with all the chemicals for antiseptics and disinfectants. So I took this out, make it a little easier on you. I also went through and made a lot of practice questions for you. So please take the time to answer each of these, check your understanding. I wasn't able to continue the numbering between slides. So what I did was I used numbers on one, letters on the next, and then back to numbers, and then back to letters. And then that's it. So you have for this whole chapter, you have lots of practice questions. Please, again, go through these practice questions and then confirm your answers with me during office hours. All right, so that's it. We're done. Make your flashcards or however you study. Start memorizing lots of info. You can do it. All right.